Well, hello everyone and welcome. My name is Marie Johnston. I'm the Education Project Manager and Content Creator at the Soil Science Society of America. It is my pleasure to introduce all these wonderful people around me. And this is a joint webinar entitled Soils and Climate Change. And it is the first event to be held together by the Soil Science Society of America and the Soil Science Society of China. So just one slide to show you here. Please contact my colleague Lacey Edwardson if you experience any technical issues. Her email is on the screen and I'll add it to the chat here in a minute. Please note that this event is being recorded, so you will receive an email when that recording is available to watch online. Again, thank you to both SSSA and SSSC for planning this live event. And at this time, I would like to introduce our first moderator. Her name is Dr. Feng Wang. Dr. Wang is a professor at the Chinese Academy of Sciences, and her research focus is soil pollution and remediation. She is the chair of the International Collaboration Working Committee of the Soil Science Society of China and the co-chair of the joint working group who organized this webinar. So welcome, Dr. Wang, and take it away.
Thank you for Marie's introduction. Good morning, dear professors, students. Good morning from China and good evening from US and also maybe some scientists from other countries. Welcome to join this Soil Science Society of American and Soil Science Society of China joint webinar on soils and climate. Actually, we have a series of different topics of seminar on joint organized by the two societies. And today is the first seminar, which is topic on the soils and climate change. So before we start, I would like to briefly introduce all our distinguished guests on the panel. Professor Dr. James Tida from Michigan State University. Professor Xiaojuan from Institute of Soil Science, Chinese Academy of Sciences. And he's also the Executive Vice President and also General Secretary of the Soil Science Society of China. Professor Michel Samson from the University of Ohio, and also he's the elect President of the Soil Science Society of America. Professor Sabine Grunwald from University of Florida, Professor Rattan from University of Ohio State University, Professor Jie Zhou from Nanjing Agricultural University, and Professor Xiaojuan Feng from Institute of Botany of Chinese Academy of Sciences, and also Professor Robert Hong from Iowa State University. So welcome. So now it's my great honor to host this session and like to invite Professor Michel Samson to give opening.
On behalf of the Soil Science Society of America, I am delighted to welcome each of you to this webinar. It's co-sponsored with the Soil Science Society of China, both societies working together. It's an event that is the first of what we hope will be a succession of collaborations between the two societies. There are so many things that our two scientific societies have in common. We represent diverse soils and diverse services that soils provide to human societies. We represent strong scientific and academic institutions that develop and publicize soil science. Both of our countries have strong agricultural production, and we also have environmental issues that are related both to agriculture and to industry. So we both need to find the best ways for soil management to help in the international effort to mitigate climate change that is relentlessly bearing down on our planet. We're all charged with the task of wise stewardship of our soils and of the planet. We know that human health, food security, and mitigation of climate change all depend on soil security and environmental quality. These are things that we are all passionate about. So one small step that we can take is to share our knowledge. We can connect scientists and teachers and students with one another, and we can preserve and improve the channels of communication. So welcome to this webinar. I am very glad that you are able to join us on this step in a long, fruitful journey together. Welcome.
Thank you. Now let's invite Professor Xiaojuan, the Vice President of Soil Science Society of China, to give us the welcome remark. Please, Professor Yuan.
Dear Professor Mike Thompson, President-elect of the Soil Science Society of America, dear distinguished professors and guests, ladies and gentlemen, on behalf of Soil Science Society of China, I welcome you all to join this webinar on soils and climate change, co-organized by Soil Science Society of China and Soil Science Society of America. First and foremost, I would like to take this opportunity to thank the organizing committee of this webinar, led by Professor James Tida, Sabine Grunwald, Michael Thompson, Jianwen Zhou, among others. Climate change is the greatest threat to the natural environment and human society that the world is facing today, and its effects are being observed globally in various ecosystems. Soils play a vital role in sustaining life on Earth and are also a direct victim, driver, and regulator of climate change. Although soils can act as a sink of greenhouse gases that contribute to climate change, our activities that exacerbate soil degradation, desertification, and pollution leave our soil systems vulnerable to climate change and impede our progress toward sustainable development. To ensure the continued existence of humanity on Earth, all countries must work together to manage our soils quickly and effectively in a way that will counter the threats posed by climate change. In 2021, China and the US committed to working together and with many other countries to find solutions to climate change, which requires immediate and thorough action. As a following step, we established collaboration between the Soil Science Society of China and the Soil Science Society of America last year in an effort to foster international collaboration and academic exchange. As the world faces the challenges of food security, climate change, and environmental management, there's no doubt that this platform will act as a channel for the transfer of knowledge and experience in harnessing soil science to sustain soils for our common future. In view of the research potential of our two countries, I'm optimistic that the information we will share in this webinar will enlighten our community and spark interest in further research on the management of global soil systems in the face of climate change to ensure a better future for ourselves and future generations. Therefore, I encourage you to take full advantage of this opportunity to listen to the groundbreaking research presented by soil scientists from both China and America. Thank you.
We're closing the ceremony here. Now we're going to the presentation session. So I'd like to invite Professor Jianwen Zhou to host this session. Professor Zhou is an outstanding Young Scholar Awardee from the Natural Science Foundation of China. He's a very famous professor from Nanjing Agricultural University. So, Professor Zhou, please.
Okay, good morning or good evening, everyone. Now we move on to the presentation session. Today we will have 4 professors to present their works on the topic of soils and climate change in this meeting. Ideally, each speaker has 20 minutes for presentation and 5 minutes to take questions. At the end of 20 minutes presentation, we may appear on screen as a reminder to end your talk soon. Now the first speaker goes to Dr. Rattan. Dr. Rattan is a distinguished professor of soil science at the School of Environment and Natural Resources at the Ohio State University. He is a former president of both the Soil Science Society of America and the International Union of Soil Sciences, and a laureate of the World Agriculture Prize. His signature research areas include soil carbon sequestration and climate change, soil health and restoration, tropical systems, and conservation agriculture. Today, his presentation focuses on managing soils for adaptation and mitigation of climate change. Now, please welcome Dr. Lei to start his talk. Dr. Lei, please.
The slides up. Thank you. I hope you can hear me and you can see the slides. It's a great privilege and honor to be invited, and I will stay within the time I'm given. I think I'll be focusing more on the processes of carbon sequestration in soil, and for the end, say a few comments about the private sector and what could be done. Just to give a background, the carbon concentration in the atmosphere, total stock, pre-agriculture, about 10,000 years ago, was 360 petagram, gigaton, billion tons. In 1750, at the pre-industrial revolution time, it had already increased by 200 gigaton to 560. At present, it's about 880 gigaton. So the difference, 560 to 880, is due both to fossil fuel combustion and land use change and agriculture. Total emission from land use change and agriculture to date is 575 gigaton, and total emission from fossil fuel since 1750 is about 445 gigaton. So agriculture has been a major source of the atmospheric CO2 concentration, which in 1960s before was 315 parts per million. 1st of January 2020, 416. 22, 1st of January, 418, and 15th of May, it reached 420 parts per million. At present, it's about the same, 420 parts per million. The question that policymakers have to address, that the total amount of carbon that humanity can emit, maximum, let's say, of 560 parts per million, minus 420 parts per million today, multiplied by 2.13 gigaton parts per million, that comes to about 298 gigaton, not 320, 298. So how do you divide this carbon pie amongst the nations of the world? That brings us to understand the processes of coupled cycling of water with carbon, with nitrogen, phosphorus, and sulfur. And it is this coupling that generates essential ecosystem services, including food security, water quality, biodiversity, and other. So looking at the soil part, which has 2 distinct components of carbon, organic, the dark material in the surface horizon, and in some soils, especially of the arid region, the inorganic, the lower material, that lighter color. And soil together, organic and inorganic, is the largest reservoir of carbon, plant nutrients, fresh water, and biodiversity. And the interaction between pedosphere and other ecosphere, of course, is the basis of life. Coming to the organic carbon, or the total carbon pool, the total carbon has the organic component and inorganic component. Organic different types, live, undecomposed, and decomposed material. The decomposed can be protected with clay organomineral complexes and unprotected, and the unprotected may be dissolved organic carbon, particulate organic carbon, macroorganic carbon. Now, the link between inorganic and organic is through the decomposition that leads to release of CO2 in the soil, and that which dissolves with water forms dilute carbonic acid, which reacts with potassium, calcium, magnesium, lead to the formation of pedogenic carbonates, which are carbonates which we call secondary carbonates. They are carbon sequestration as secondary carbonates as soil inorganic carbon. They are distinct than the lithogenic carbonates. And the pedogenic carbonate might be both carbonates and bicarbonates. The weathering of silicates, which is a long-term geological process, but some minerals such as olivine might decompose quicker if they are made into powder. The reaction is simple, leading to formation of calcium and magnesium carbonate. In some profiles, you see the secondary carbonate as a sheet or as granules, as you can see in this slide. And this process is very much prominent in arid climate, especially if the management is good. I share with you some pictures from China with a rainfall of about 250 to 300 millimeter, where with gravel mulch and drip irrigation, as you can see both in this slide, very good crops can be obtained because of the better management. And this management does lead to both sequestration of organic and inorganic carbon. So melon, here is sunflower, here even the wheat. And the gravel mulch obviously stays there for maybe a decade or longer. It doesn't need to be renewed very quickly. Now coming to the organic carbon part, which is very important in humid soils, and go back to what Charles Kellogg of USDA said, essentially all life depends on the soil. And there can be no life without soil and no soil without life. They have indeed evolved together. I would take that statement one step further and indicate that rhizosphere is the only place in the universe where the death is resurrected into life. So soil has that capacity to transform dead material into life. And from that point of view, many people think soil is indeed a living entity. And that living entity in life support processes are primarily driven by soil organic matter content. The soil organic matter content is its dynamics that creates water resources, quality and quantity, climate change mitigation, adaptation, stabilization, food security, both in terms of quantity and quality, and of course the biodiversity. Therefore, soil organic matter content is the heart of soil health. And that's the focus we want to make here today. So when we transform the natural ecosystem to agricultural ecosystem, such as in the Midwest United States, Ohio, for example, Michigan, other states in the region, the soil organic carbon stock can decrease to 50% within about 50 years. After that, if the soil erosion continues, the organic carbon stock can continue decreasing. But if erosion is stopped by management practices, it can reach a stable equilibrium. At that point in time, if we adopt better management practices, which vary from place to place and soil to soil, it is possible to increase the organic carbon content following a sigmoid curve. And that sigmoid curve can again stabilize after about 25 or 30 years or longer to a level which we can call attainable potential. At that point, if another technological option innovation can be adopted, eventually in 2 or 3 generation period, 50 to 75 years, it is possible to go back to the original level if the soil management was done properly. Many times people ask the question, can you go beyond the original level? Not very regularly. In some cases, where the native systems have some constraints, water, phosphorus, maybe acidity or other limitation, it may be possible to go beyond the initial level. By and large, such as the soil we have in Midwest, no. The original level is the maximum. And I think if we can reach even 66% of the original level, we have done quite well. This kind of graph, which you can get from long-term research, if you take the slope of the line, you get the rate of carbon sequestration. You get the carbon sink capacity from where we were and where we can be with long-term management. We can calculate the mean residence time, which is pool over flux, and identify the technological options. There's not one, there are many. For 300,000 known type of soil series, it's not possible that there's one option. Option examples include conservation agriculture, biochar, agroforestry, desertification control, afforestation, pasture management, water harvesting, drip fertigation, farming system, and many others. The important part is never to forget that each of these practices have their own cost of doing business in terms of the carbon and water footprint. Therefore, from the gross rate that you get from delta Y over delta X, we must subtract the amount of carbon used to produce those ecosystem services. In terms of what happens, here is a crop residue, which if it's put back on the land, it goes through biochemical transformation to become humus. And as this transformation happens, there is additional nutrients required. Why are the additional nutrients required? Because the carbon to nitrogen ratio in crop residue of cereal is about 100. That in humus is about 10 to 15. Carbon to phosphorus ratio, 200 residue, in humus about 50. Carbon to sulfur ratio. So this can depend on the sustainable management of soil. Many people ask, what is sustainable management? We must replace whatever is removed. We must respond wisely to what is changed. We must predict what may happen from anthropogenic and natural perturbation. And somehow or the other, we must produce more from less. We have been, in general, using a lot of inputs, lot of land resources. Hopefully, we can save some land for nature through adoption of nature positive soil management practices, such as protecting, managing, restoring soil, strengthening biodiversity, improving water quality, adopting negative emission farming. I'll come back to that in a moment. Reducing input of chemical, using biostimulants and organic amendments, promoting a biocircular economy, supporting the One Health concept, health of soil, plants, animal, people, ecosystem, and the planet is one and indivisible. And using digital innovation wherever possible. One of the examples which is commonly taught these days is regenerative agriculture or agroecology, also called very popularly, especially in Europe. Both of these concepts refer to a strategy, an approach, an idea. These are not a practice. There are many practices which may come under this category. So what is this regenerative agriculture? It's an eco-innovation. It's powered by known carbon energy. It's driven by a circular economy and green infrastructure. It's supported by the recarbonization of the terrestrial biosphere. Remember, I mentioned we lost more than 500 gigaton from land. We must put it back. That is the basic bedrock of sustainable development, recarbonization of the terrestrial biosphere. So the question is continuous soil cover, no soil disturbance, integrated nutrient management, complex crop rotation, which can lead to positive soil carbon budget. And this is what we like to call carbon farming. What does carbon farming mean? It involves management of carbon pools, flows, and greenhouse gas fluxes at farm level with the purpose of mitigating climate change in such a way that we can mitigate the greenhouse gases, but the farmers can get some income, just as they get income from selling wheat or corn or soybean or milk or poultry. They can also get income from selling carbon. And that carbon stock can be improved by creating a positive soil carbon budget. From that point of view, soil can be a bank account, similar to the bank account. If you want your bank balance to increase, you must put more into the bank than you withdraw. In soil, the same thing. If you want your carbon budget to increase, you have to put more than you withdraw, and more can be by many practices, which I indicated before, and they must be balanced in a positive way against losses by erosion, leaching, and decomposition. Unfortunately, in many cases, especially the traditional small landholder farmer in the developing world, the gains are always lower than the losses. Therefore, we are always losing carbon stock from soil. In many cases, the good practices are better, which I will not discuss again those, but the rate of carbon sequestration generally is about 400 kilogram per hectare, such as in the Indo-Gangetic Plain, in sub-Saharan Africa, where I did this kind of research, it may be about 500 kilogram. It's more humid where I was, but global rate have many times been reported somewhere about 500, 600 kilogram of carbon per hectare per year. The question that many times people ask, how much carbon can you put back in soil? Generally under ideal conditions, and we are no longer near ideal, I don't think we are doing very much positive balance, which I showed on a global scale. But if we did, the rate maximum is about 2.5 gigaton of carbon per year. Remember, what we emit from fossil fuel is 10 gigaton. What we emit additional is 1 gigaton from land use conversion. So out of 11 we emit these days, 2.5 maximum can be put back in soil as organic. There is some additional inorganic. Several colleagues of us got together and calculated what can happen between 2020 and 2100. The soil carbon stock we can put back about 180 gigaton, vegetation about 150, total globally technical potential about 330 gigaton is equivalent to drawdown of atmospheric CO2 about 150 to 160 parts per million. Therefore, if we have a policy to stop burning fossil fuel, and if we have positive policy which are pro-nature and pro-farmer to support them for land, carbon sequestration, soil and vegetation, keeping global warming to 2 degrees centigrade, not more, is still possible. Not complete, but some. But to do that, farmers must be rewarded properly. And that proper reward, according to my calculation, the cost of crop residue, the cost of NP and sulfur additional nutrient required, the land value, another $130 per ton of carbon, $35 per ton of CO2. Therefore, if a farmer sequesters half a ton, $65 per hectare, $26 per acre. If a farmer sequesters 1/3 of a ton, $43 per hectare, $18 per acre. If you don't pay that much, if you say we'll pay you $5, some company, very nice of them, they're doing that. That's undervaluing a resource which is so precious and not being given proper value. I would propose a timetable on a global scale, which is probably very ambitious, probably may not happen. But if we were to solve the problem of climate change and make agriculture a solution, we have to take some drastic steps. Fertilizer. We are using 200 million tons globally. By 2100, by improving soil health, by improving use efficiency, maybe we can aim at 50 million ton. Cropland area, we have now irrigated area 350 million hectare. We can probably improve to 500 million hectare. Total cropland, which we have 1.5 billion hectare now. If we use productivity management system that we know, especially in developing countries, we can reduce the area to half. Conservation agriculture, on the other hand, all the entire cropland should be under conservation agriculture by 2100. Grazing land, right now we have almost 4 billion hectare. We don't need that much. We can do better and we can manage within about 1.5 billion hectare. Water use in irrigation, if we use drip fertigation system, not flood irrigation, not sprinkler, not furrow, drip, we can do with 1/3 of the amount of water we use now. And yet we can increase productivity of the cereal grain from 4 ton globally now to 8 ton. Is this doable? Technically, yes. Will it happen? For that, we need education and role of private sector to translate science into action. Why? By promoting nature-positive agriculture, by increasing access to inputs, by improving investment in agriculture research and development, and by working with farmers. Cooperation with private sector can also help achieve sustainable development goal, especially number 1, 2, 6, 13, and 15, where cooperation between private sector, policymakers, academicians, national institutions, land managers together can implement the knowledge we have into action at a global scale. The last slide that I want to show you is the mantra, which I personally believe very strongly in. And that mantra is that the healthy soil equal to healthy diet, equal to healthy people, equal to healthy ecosystem, healthy planetary processes, and the global peace and stability. Thank you very much.
Very much, Dr. Lin made an interesting presentation. Thank you very much. We have several minutes to take questions. Actually, I have a question. Could you talk a little about carbon farming? In other words, how to consider the potential for farmers to store more carbon in soils by changing soil management?
Yes, carbon farming means making carbon in soil, both organic, inorganic, and in vegetation, trees, as a commodity that can bring income for farmers. How can it bring income for farmers? There are two options. One is let market forces decide what the price and demand is. We had a Chicago Climate Exchange that started in 1999. It was at $1 a ton. The market went up to $4 to $5 by 2005, 2006. And because we did not have a cap on burning of fossil fuel, the market collapsed to 20 cents. And I don't think anybody get very excited to get 20 cents per ton of carbon dioxide, when the real price, as I mentioned, societal value is $35. That was 2012 prices. The other possibility of carbon trading and carbon rewarding farmer is payment for ecosystem services. Not what the market says, but what the real societal value is. I'm hoping that Farm Bill, which is coming up in the US, if they have a Soil Health Act, similar to Clean Air Act and Clean Water Act, and have a Soil Health Act and a Soil Health Act, farmers that do good thing and enhance soil carbon are rewarded according to the societal value, $35 per ton, or even up to $50 now. That is more way to motivate the farmer. And if the US and China can have something like that, I'm sure the rest of the world will follow.
Okay, thank you. For the time limitation, we take questions here. Thank you, Dr. Lin, again. Thank you.
Thank you so much.
Now we move on to the next speaker, is Dr. Xiaojuan Feng. Dr. Xiaojuan Feng is the principal investigator and the vice president of the Institute of Botany, Chinese Academy of Sciences. He is specialized at using molecular level techniques, such as biomarkers and isotope analysis, to examine the transformation and the transport of soil organic matter under climate changes. Now, please welcome Dr. Feng to present her work on molecular alterations of soil organic matter in alpha grasslands and warming. Dr. Feng, please.
Thank you. Can you hear me?
Yes,
Can you
yes.
hear me? Yeah, okay, thank you. Thanks, Professor Zhou, for the introduction, and thank you to the organizers for this opportunity to share some of the recent work that my group has done. And I feel honored to speak after Dr. Lau, who gave a grand and excellent introduction of soil carbon cycling and in terms of combating climate change and the overall health of ecosystems. And I think I'm going to talk also about soil carbon cycling, but I'm going to showcase several case studies related to soil carbon response to warming in the alpine grassland. Some background information. Alpine ecosystems store huge amounts of soil organic carbon and are sensitive to warming, therefore attracting enormous research attention. Such ecosystems are distributed at high latitudes and altitudes, and the Qinghai Tibetan Plateau, or what we call QTP, harboring vast areas of alpine grasslands, is one of such ecosystems. And in the past few decades, Qinghai Tibetan Plateau is experiencing a warming trend that is much faster than global average, inducing dynamic changes in the vegetation distribution and phenology, also known as greening of the plateau. So how does warming-induced vegetation shifts affect soil carbon processes and dynamics underneath? My group and I try to address this question using molecular level techniques, including biomarkers and compound-specific carbon isotopes. So our research was based on the Haibei Research Station located at the northeastern corner of Qinghai Tibetan Plateau. It has an elevation of 3,200 meters and a typical vegetation coverage of relatively moist alpine grassland of quite high plant diversity. In 2011, Professor Jingshen He from Peking and Lanzhou University started a climate control experiment at Haibei, consisting of warming treatment by infrared ray heaters from above ground, as well as precipitation management, including 50% less and 50% more precipitation as compared to the regular control. And each treatment had about 6 plots. This picture shows the temperature contrast at the soil surface between the warming and control treatment. On average, regular warming increases the soil surface temperature by 1.8 to 2 degrees C annually, and we call that W1 treatment. We also have a W2 treatment, which is winter-enhanced warming, that increases the soil surface temperature by about 3 degrees C during the winter from late October to late April, and the increase was less from 0.5 to 1 degree C for the rest of the year. But on average, both W1 and W2 warming treatments have similar magnitude of warming on an annual basis. So the warming effect is very evident, especially in the winter for the W2 treatment, and you can see the snow is gone under the heating plot. And as I just mentioned, the alpine grassland at Haibei Station has a quite high plant diversity, and the dominant species can be grouped into 3 categories: sedge, forb, and grass. Compared to sedge and forb, the grass species have deeper root distribution, which enables them to access the deeper soil moisture, especially during drought. And coupled with about 32 years of ground monitoring of vegetation composition, Professor He's group found that warming and drought enhanced the competitive advantage of deep-rooted grass species, leading to a shifted composition of the local vegetation and increased root biomass in the deep soils. So the question that follows is, how does deep root growth affect soil organic carbon dynamics under warming? Will warming increase the sequestration of recent photosynthesis from plants and at depth, or will it activate soil microbes underneath and prime the degradation of the native, already sequestered soil organic carbon? And if both processes or either processes happen, will that lead to a shifted composition of soil organic matter? And to answer these questions, we sampled soils from the top 0 to 10 centimeter and subsoil 30 to 40 centimeter before and after 3 and 5 years of warming treatment from the Haibei Station. And as you can see here, bulk SOC did not change at either depth after 5 or 3 years of warming. We then utilized biomarkers of both plant and microbial origins to glean on warming-induced shifts in the soil dynamics, sources, and processes. And these biomarkers include plant-derived lignin phenols, which is a major component of plant litter and a contributor and an input of soil organic matter. And less abundant biomarkers include plant-derived wax lipids, cutin, suberin, which are monomers of the protective coatings of plants at the surface. We also include microbial-derived biomarkers, including the biomass indicator PLFAs and necromass indicator amino sugars. Amino sugars are a cell wall component of microbes, and they accumulate in the soil even after cell death. So they are commonly used as a biomarker for microbial necromass. We also analyze some other group of microbial membrane lipids, GGGT, which is different from PLFA. Anyway, we analyze these different molecular components in the soil and compare them relative to the control plots. And as you can see here, relative to the control, both W1 and W2, the regular warming and winter enhanced warming, decreased phenols that mainly derived from plant lignin in the subsoil, but increased microbe-derived lipids as well as sugars of both microbial and plant origins. However, none of these changes were observed in the topsoil. Moreover, we use soil fractionation and C14 analysis and show that both the organic carbon content and C14 content increased in the minerals associated fraction, but not aggregates, including macro aggregates and micro aggregates in the subsoil. This suggests an accumulation of new carbon in the mineral-associated organic matter, MOM, in the subsoil. Further biomarker analysis of the soil fractions revealed that lignin phenols were relatively concentrated in the aggregate, especially macro aggregate fraction, while sugars were more concentrated in the small aggregates and mineral-associated fraction. So collectively, these results suggest that warming induced an enhanced decay of lignin that mainly resided in the macro aggregates in the subsoil, but induced an accumulation of new carbon in the form of sugars and microbial lipids in the mineral-associated fraction. So why was lignin more vulnerable to decay at depth under warming? We conducted a priming experiment in the lab using C13 labeled glucose to simulate increased plant root exudates in the soil under warming. We found that glucose addition induced greater microbial priming of the native soil carbon in the subsoil than the topsoil, and greatly increased lignin solubility in the subsoil at the same time. Hence, the deeper root growth under warming primed soil microbes and enhanced lignin degradation, probably because lignin was not firmly associated with minerals and hence not protected by the minerals. Now that soil microbes were activated by root growth at depth, we wonder if microbial residue accumulation also enhanced in the subsoil. Our soil lipid analysis I just showed now definitely suggested so, because we observed a relative increase of microbe-derived lipids, including GDGTs and other monomers in the subsoil. But we did not observe an increase of microbial biomass in the soil at depth. So why? Well, microbial production of residues or microbial carbon is not only related to the size of the microbial community, but also fueled by labile carbon or plant photosynthesis. In other words, the soil microbial carbon pump, which is a very popular term now, is ultimately driven by plant inputs, and hence the accumulation of microbial carbon should be linked to primary productivity. Warming-induced plant growth, as we observed at Hebei Station in the deep soils, likely fueled microbial carbon pump therein and led to microbial residue increase at the Hebei Station. However, as we cannot tease apart newly produced microbial residues versus the old residues that accumulated over time in the soil, we cannot directly verify the link between increased root biomass production and microbial residue accrual at the high base station. So we leveraged a large-scale investigation on microbial residue distribution in Chinese grasslands to show primary productivity's influence on microbial necromass accrual in the soil. For this, we sampled surface soils on the Qinghai Tibetan Plateau as well as in Inner Mongolia, spanning a wide range of grasslands with different vegetation types, climate, and productivity. We also synthesized amino sugar data from globally distributed grasslands for comparison, which is a biomarker for microbial cell residue, as I just mentioned. We see that the Qinghai Tibetan Plateau grasslands have higher SOC content than Mongolian temperate grasslands, but lower content of microbial necromass relative to SOC, despite similar amount of microbial biomass in the soil compared to all other grasslands. Further, using a structural equation modeling approach, we show that microbial necromass accumulation quotient, which is a ratio of microbial necromass carbon versus microbial biomass carbon, is constrained by both moisture availability, i.e. aridity index, and net primary productivity, NPP, across the QTP and Mongolian grasslands. So for microbial community of similar sizes, soils in the moist and more productive regions contain more microbial necromass contribution. This result demonstrates that plant productivity has a large effect on the microbial necromass accumulation in the grassland. And hence, stimulated plant growth may promote the accumulation of microbial residues at high base station under warming. So if the plant growth is stimulated and necromass production process is also enhanced, will this lead to increased soil organic carbon content or stocks in the future? I think this is the ultimate question that many people are interested in. Well, before answering that question, I need to introduce or define a term, the microbial carbon pump efficiency. As we just mentioned, the microbial necromass production is accompanied by respiration and is fueled by plant carbon inputs. So we should not only care about the total amount or content of microbial necromass residues in the soil, but also about the efficiency of the microbial conversion process from plant carbon into microbial residues. How do we assess this? Well, we know that microbial carbon use efficiency, CUE, can be used to evaluate microbial efficiency of biomass production relative to respiration. However, only a small fraction of microbially synthesized carbon can be preserved in the soil after cell death. And this preservation process is probably mediated by mineral protection and other abiotic processes, so it can be decoupled from microbial physiology processes. So the CUE may not be a very good term to determine the efficiency of the microbial carbon pump. And hence, we need a better term to assess the overall accumulation of microbial residues relative to respiration. For this, we use amino sugars as the microbial cell residue marker. And we introduce this term, which is amino sugar accumulation efficiency, AAE. So we first use a model soil incubation experiment to simulate the accumulation process of microbial residues. And we want to show that the accumulation efficiency of total microbial carbon, biomass, and amino sugars may be decoupled. We incubated grassland soil inoculum in artificial soils containing different levels of bentonite or ferrihydrate, fed the microbes with fructose that is not synthesized by microbes, and incubated the soils for 4 months. During the process, we measured respiration, PLFA, which is a biomass indicator, amino sugars, an indicator of the necromass, and calculated total microbial carbon by subtracting fructose remaining in the system, which is the sole carbon source or substrate source for the microbes. And this figure shows how microbial carbon accumulated and respiration increased as fructose was used. We calculated the accumulation efficiency of total microbial carbon, which we call CAE, amino sugar accumulation efficiency, AAE, and compared with microbial respiration quotient, QCO2, which is a reverse of CUE. These variables show different variation patterns in the soils of varied mineral content, and they were only related in the soils of highest clay content. Hence, we show that microbial carbon efficiency may not be accurately tracked by CUE, and amino sugar-based accumulation efficiency may be a better term to determine this variable. So next, we use this variable to examine if soil microbial conversion efficiency increased under warming, drought, and increased root inputs at the high base station. We collected soils after 5 years of manipulation experiment and incubated the soils with C13 labeled grass roots and leaves. We measured PLFAs and amino sugars derived from the newly added litter after 3 months of incubation. We found that both CUE and amino sugar-based AAE decreased with drought and warming treatment in the subsoil, but not topsoil. At the same time, 5 years of warming increased subsoil nitrogen limitation by decreasing inorganic nitrogen supply relative to water extractable organic carbon in the soil. So possibly because warming also enhanced the microbial versus plant competition for inorganic nitrogen in the soil. The variation of AAE in the subsoil was closely related to the degree of nitrogen limitation. And hence, although microbial-derived components showed a relative increase in the subsoil after warming, the efficiency of microbial conversion processes decreased due to nitrogen limitation. So microbes consumed more plant carbon and likely produced more CO2 as well. So back to the question, will SOC content increase with continued warming at the high base station? Well, my guess is probably not. Although we see increased buildup of microbial residues and increase of the mineral-associated fraction in the subsoil. So warming will probably shift the composition of the soil organic carbon pools, but the increase of the mineral-associated fraction comes at a cost of increased plant carbon consumption as well as native organic carbon priming. I think the future or the likely increase of soil organic carbon very much depends on how nitrogen supply meets the needs of the microbial community, therefore not constraining the efficiency of the microbial carbon pump. Okay, finally, to conclude, warming-induced vegetation shifts at the high base station increased deep soil carbon input, primed lignin degradation, and increased microbial carbon accumulation, but not in the topsoil in this alpine grassland. However, the microbial carbon accrual increased at a cost of reduced microbial carbon pump efficiency due to nitrogen limitation under warming and drought, suggesting that subsoil microbes were less efficient in preserving plant-derived carbon. And hence, the accrual and stability of subsoil carbon pools in alpine grasslands depends on the interactive effect of carbon supply and nitrogen supply in the future. So with that, I want to thank you for your attention, and I welcome any questions.
Okay, thanks, Dr. Feng. A very good job and an interesting story about the soil organic carbon response to warming. Any questions? Okay, actually, I have a question. how do you expect the observed soil organic carbon compositional changes linked to soil organic carbon stock variations in the future?
Yes, I briefly mentioned, I think so warming definitely through inducing deeper root distribution shift the organic carbon dynamics and sources. So in the future, I expect at the high base station, we will see more mineral-associated carbon accumulating in the subsoil, but this process will probably consume the macro aggregates or micro aggregates, so the carbon not protected by minerals. So in total, there probably will be a balance or even a decrease of total soil organic carbon stocks, but we will see a shift in the composition of the stock of the carbon pools.
Okay. Also, you mentioned the macro. Do you think that the new carbon accumulated in subsoil mainly comes from microbes?
I think there must be a microbial contribution because we do see some of the lipid markers derived from microbes increased in the subsoil. But I don't think all the new carbon that accumulated in the subsoil comes from microbes. I think there are some untransformed plant molecules accumulating there as well, because we see a reduced microbial conversion efficiency. And I think some of the new carbon may not all go through the microbial conversion process, the microbial carbon pump.
Okay, thank you again. Thank you very much. Good very job. Okay.
Thank you.
Now we move on to the next bit. We invite Professor Robert Rattan to host the following meeting time. Dr. Rattan, please.
Thank you. So our next presenter is Dr. Sabine Grunwald, a professor of Pedometrics, Landscape Analysis, and GIS at the University of Florida. She's an SSSA fellow and internationally recognized for research in pedometrics, carbon science, and soil health, focusing on digital modeling of soil and terrestrial carbon from field to global scale, modeling of soil ecosystem functions, soil proximal sensing, remote sensing, geoscience, and AI applications to soil health. The title of her presentation this evening, or this morning, wherever you are, is Data and Sensor-Driven AI Modeling of Soil Carbon from Local to Global Scales. Dr. Grunwald.
So this talk focuses on soil carbon modeling from the perspective of AI modeling. And I have the pleasure to follow up on the talk also given by Rattan Lal that touched also very briefly on some of the material that I like to cover in more detail. First, when we think of soil and climate change, the question about optimization comes to mind. Optimization of soil carbon sequestration and the reduction of land-based greenhouse gas emissions. That is critically important for soil health, but also food security and carbon accounting and marketing, as well as resilient agroecosystems. So the question about whether soil has acted as a sink or source of carbon depends on many different factors that vary spatially and temporally. So this question is not that easy to answer, in particular looking at global or national scales. So the presentation will focus on particular data and sensor-driven approaches using AI, machine learning, and deep learning approaches, focused on soil carbon storage and soil respiration. I will present also a few examples from Florida, in the United States, and also global mapping applications. So when we think about the optimization of carbon for carbon farming and marketing, it depends on attaining the maximum attainable carbon storage in a soil. But there are, of course, technological potentials through technical interventions or specific amendments that can boost up also the carbon gains in landscape. Of course, there are carbon losses and even leakages by residue removal, for example. And of course, the question about carbon avoidance, the reduction of greenhouse gas emissions, in particular the trace gases, CO2, CH4, and N2O. But when it comes to carbon sequestration, in particular at a global scale, for example, global croplands, these numbers that have been assessed vary widely. Here by ZOMA, an aggregate number between 0.9 and 1.85 petagram carbon per year. This amounts about to 26 to 53% of the target of the global 4 per mil initiative that was globally also put forth as part of COP21. But talking about carbon budget means also we have to address issues of the quantification, in particular spatial and accurate characterization of soil carbon stocks and sequestration rates, to address uncertainties in modeling, to scale up field to global scale. That includes also to address issues of data currency and density, and of course to consider also the geographic variability of soil and environmental factors. Now, with the emergence of AI modeling approaches, there are capacities to form big data hypercubes that include soil measurements, but also hundreds and hundreds of environmental covariates that characterize an ecosystem, that are derived using remote sensing techniques, but also proximal sensing techniques. And these type of measurements can characterize the space and the temporal components of ecosystems contiguously, but also with finer and finer spatial resolutions. This has enabled the emergence also of AI using pattern recognition, learning algorithms, hyperparameters, latent variables to develop digital soils or digital soil carbon stocks and carbon footprints. Of course, these type of models differ from process-based models that are typically developed on site-specific, plot-specific, or in laboratory settings. Now, when we talk about AI, what exactly is that? AI has been characterized as machines, intelligent entities that have the capacity also to provide predictions or capacity of knowledge to address novel situations. Machine learning refers to machines or systems that can learn from experience, in essence, empirical data supplied and algorithms that train models in a calibration phase followed by a validation phase. These machine-driven recognition of pattern and structures really are brute force fitting between input and output variables. Deep learning is very similar. However, it introduces multiple processing layers to learn more complex high-order relationships that also have some level of abstraction. These DLs are relatively new. They have emerged over the past few years. But still, DL, deep learning, is still just another statistical learning approach that extracts these features from raw data sets. Now, when we think of ecosystems, agroecosystem, forest ecosystems, natural ecosystems, We can characterize different components, soil topography, ecology, parent material, or the geology, atmospheric variables, for example, climatic parameters, water, biota, for example, vegetation, land use, but also management and also human interventions or human management that is imposed on a landscape. Now, when we talk about these broad categories, they can be populated by hundreds of variables that are derived from sensing, but also from measurements or databases. Now, in addition to soil carbon measurements, the AI models allow, for example, training of these models and then the assessment or the validation using independent data sets. Now, just a brief glimpse into a few of these AI algorithms. So in essence, when we think of the circle, the step-up circle, these are the input data that put into these type of regression tree models. These are ensemble regression trees with hundreds and hundreds of input variables, but also hundreds and hundreds of iterations or runs to make predictions, for example, for soil carbon or other soil health properties. But we see here some of these AI methods have become very sophisticated and advanced. For example, convolutional neural networks that allow the processing of input variables going through different steps of convolution, pooling, flattening, and reconnecting layers to make the final predictions. There's a lot of abstraction and sophistication involved that actually the machine performs. Now, what are some of the results related to AI-based soil carbon modeling? At global scale, several soil organic carbon maps were generated using AI technology. Here from soil grids, we see that this map was derived using over 240,000 locations globally and over 400 environmental covariates of the circle representing really different ecosystem factors. The AI method quantile random forest was used. What stands out is, of course, the concentration of higher values of carbon in the higher latitude areas, but also in the mountainous areas that were mapped out here at 250-meter resolution. Now, in terms of the cross-validation results, the root mean square error was about 40 decigram per kilogram compared to the range of values up to 5,500. This is actually a quite small value. However, we see also that the uncertainty of some of these predictions were more limited. In particular, the lighter colored areas show also more uncertainty. And this gives us also a pause because, of course, more data would result into also better predictive capabilities. Now, in the US, we produced a soil organic carbon map topsoil using topsoil measurements and profile measurements between 0 and 100 centimeter. You see here the distribution of the measurements, not even, it was slightly unbalanced. We used, in this case, AI machine learning Baruta in combination with random forest modeling and splining of the vertical profiles. Over 100 environmental covariates were used for the predictions. But the question is really, what type of predictors served to explain some of the soil organic carbon stocks in the United States? And these were soil properties from databases here mapped out in brown color. Also, hydrologic properties had a tremendous capacity to infer on SOC, ecology. We see climatic variables. A few of them were selected as predictor variables by the model. Geology, parent material. Then we see also vegetation properties, biota, and finally also some topographic variables. But what stands out is that it's a very complex, really combination of factors that can explain the spatial variability of soil organic carbon stocks in the US. Here are some maps, topsoil and the profile map. The calibration of these AI models were very, very good with an R-square of 0.92. In terms of independent validation, the models degraded slightly, as we see here, in terms of the error metrics and the model fit. But important here, these type of maps provide us really with important information about the landscape. In Florida, we also used an AI modeling technique that included 200 environmental covariates, measurement of soil organic carbon bulk density, and also various data reduction techniques to avoid also redundancy of environmental covariates. They included all relevant variable selection using Baruta and random forest, and minimal optimal selection of various techniques that were tested, and predictive modeling was conducted, testing 4 different AI methods. Now, this is a landscape that is relatively rich in carbon. It has a mean value of close to 5 kilograms per square meter. Now, in terms of the modeling results, what stands out was that some of the best models achieved an R-square of 0.63. Compared the predicted values versus the observed values, we see also that most of these models performed very similar in terms of their capacity to predict soil organic carbon in validation mode here in these graphs. When we zoom in and take a look at the cross-validation results, we find that the greedy forward method achieved nearly similar, also good predictive capabilities, however, with only 4 different variables: land use, vegetation type, available water capacity in the topsoil, and soil moisture. These were some of the main predictors that stand out. Now, of course, there were other predictors that also explain some of the variability, but climatic variable in this type of landscape was not one of the high-ranked predictor variables. We see the distribution of soil organic carbon across the state of Florida from one of the best performing models. Unfortunately, some of the Everglades area that contains actually a large amount of wetland area, these data were not available to us for modeling. Now, when we look and compare these type of models to measurements that were conducted historically, dating back to the early 1965, to a more current data set dating back 10 years ago, stretching over a 40-year period. We conducted also an analysis matching up different sites, some of them that has not changed land use, like that stayed in crop use or stayed in pineland land use. Interestingly enough, here we found that by increasing temperature, the soil organic carbon sequestration rate was quite scattered, had a slight upward trend. The opposite trend was found in terms of precipitation. Now, these results might sound surprising. However, the caveat is that this landscape also is quite complex. It is hydrologically manipulated. Agriculture is conducted on organic soils, but if you look at the land use change, the agricultural pattern over the 40-year period declined. So did upland forest and rangeland, and there was a slight increase actually in terms of wetlands. So overall, the soil organic carbon sequestration rates in Florida were governed and controlled mainly by land use changes, and only partially controlled by climatic factors interacting with land use and soil and topographic and other, in particular, hydrologic variables. Now, to take this one step further, we also took a look at carbon pools, in particular, recalcitrant carbon, a labile or hot water extractable carbon, and total carbon that was measured at 1,000 sites, also in Florida. There were environmental predictors implicated in this study, over 320. And a variety of AI machine learning methods were tested compared to each other. And also geostatistical methods, ordinary kriging was used. However, such methods were overpowered by the superior performance of AI models. Here are some select results indicating also the prediction of total carbon was superior with the random forest AI model. Now, in terms of predictors, biotic and hydropedological factors explain most of the variation in terms of carbon pools. Lithologic and climatic factors showed some moderate relationships to soil carbon and total carbon. Here are some maps. It's quite evident that total carbon and recalcitrant carbon showed very similar patterns in terms of the spatial distribution. Whereas labile carbon that is more closely tied to management showed a quite distinct different really distribution in terms of the high and low HC values, the labile carbon. Now, overall, in terms of, we see here the predicted values to the site-specific observations or measurements overall mirrored each other, although not closely, but were in similar range, in particular, a good match here for labile carbon. Now, when we take a look in terms of prediction is one thing to look at carbon budgets, but what is really of interest is the efficiency of soil carbon sequestration. So here we took a slightly different approach. Our objective was to compare site-specific soil carbon sequestration to optimal, attainable soil carbon sequestration. In essence, trying to assess what's possible. Here we coupled AI machine learning with an optimization method called data envelopment analysis, and assessed different optional also settings using different frontiers or optimal states, global and local orientations that were also assessed using input and output orientation and various optional settings. But important in this study, also in similar data sets as on previous slides, were used along with 7 select environmental predictor variables that were mapped out over long periods of time. In particular, vegetation indices like the Enhanced Vegetation Index, Normalized Difference Vegetation Index, and various others. Now, in terms of simulations that provided results, we can show them here, where we have the optimal line of technology that, in essence, identifies the optimal state. But of course, sites might differ in terms of the distance from the optimal state. This is actually mapped out by this type of DE analysis. Now, soils, of course, that showed in Florida in terms of site-specific efficiencies differed quite tremendously from here the areas that showed high efficiency that came close in terms of being high in carbon, and those areas in blue showing also low efficiency. Now, we can target these specific areas very tightly also to management and optimize also soil carbon storage. Briefly, in terms of AI spectral modeling, that has really huge potential to help us with the assessment of predictions of soil organic carbon at global scale. Here, our global study that showed an high predictive capability from proximal soil sensing data close to an R-square of 0.9. Such also results were produced using US-wide data, over 14,000 spectral scans, where one model stands out, an AI model based on Cubist, which shows an R-square of 0.95. This also tells us that the predictive capability of spectral data is superb and provides us also with fast, cost-effective, efficient methods to infer on various properties, because it does not work only with carbon, but also several other properties that are grouped under soil health. Now, when it comes to soil respiration modeling, and I'm coming here to the end very briefly, the controversy, of course, for when we look at global soil respiration is how much is it controlled by temperature and global warming? There are some studies that have shown and identified temperature as a major driver, and some studies the opposite. In essence, showing that actually biotic and abiotic properties really govern the global respiration rates. For example, 1.5 global studies. Now, it is interesting to look here at some of the AI models. It was clearly shown that AI models perform better than 10 mechanistic terrestrial ecosystem simulation models. In essence, these process models were overpowered by AI because the R-square was clearly much higher, whereas the mechanistic model showed huge variability in terms of their simulation output. Other results identified with quartile regression forest showed that climatic properties actually had good predictive capabilities to infer on soil respiration. For example, mean annual temperature, mean annual precipitation, and others. But important, other studies contradict the RS assessments, and this possibly has to do with global imbalanced data sets that are incomplete, that need also some more work to unravel also the patterns that exist between climate and soil respiration or carbon fluxes. Now, in summary, when it comes to modeling of soil carbon and other soil health properties using AI or data-driven approaches, it's a very different type of paradigm and thinking, because here the models are only limited by the data inputs. Modeling of complex, interrelated, high-order relations becomes possibilities. There are cost-effective sensing techniques available. We can create gridded data even at global scale that provide really a lot of information that is not available through soil or site-specific investigations. And also cloud computing, of course, has pushed AI also to the edge, where models can be developed at national and global scale through data integration, which requires also collaboration. Model performance is often better with AI models than any other model type. Drawbacks might be that these models are black and gray box models, mirroring the human brain. And in terms of the age factor or the human farming management data that truly are very much difficult to derive at global or national scale, have been so far marginalized in these models and need further attention to be integrated. thank you very much for listening.
Thank you. Thank you very much, Dr. Grunwald, for the excellent presentation. And you have covered all of the time frame. So no questions have come through, and so it works out perfectly. So we can move on to our next presenter. Thank you very much.
Thank you.
So our next presenter is Dr. Tida Gur, a professor of environmental soil science and biogeochemistry at Ningbo University. His research focuses on rhizosphere ecology in agricultural systems, use of isotopes, sequencing technologies, and biomarkers to evaluate soil functioning. The title of his presentation is Characteristics of Paddy Soil, Specifics in Dynamics of Organic Carbon Cycling. Dr. Gur.
Good morning and good afternoon. It's my great honor to introduce my work about the paddy. As we all know, compared with the upland, the paddy always flooded or wet and dry alternative, and this will result in the EH dynamics. So the EH dynamics will result in the specific in the dynamics in organic carbon cycling compared with the upland. So here I will focus on the microbial biological science chemical characters in organic carbon cycling in paddy soil, especially compared with the upland. So at first, I'd introduce the background. So all over about half of the world populations are the main food as the rice, and the paddy has already lasted for 10,000 years to sustainable use in the world. And as we all know, during the paddy management, there are always the wet dry alternative and the result in the anaerobic and anaerobic conditions. There's always the anaerobic and anaerobic cycling during the paddy management. And this will result in the some of the oxidation and reduction chemical cycling in the paddy, such as for the nitrate to ammonia, and iron 3 turn to iron 2, and etc. And also the paddy has more carbon sequestration potential. That's about 5 kilograms per hectare per year. So here show our long-term fertilization field experiments shows during the last 2 decades, the changes in organic carbon content of the plow layer, that means the surface soil is increased compared with the no fertilizer, both in chemical NPK fertilizer and chemical and organic fertilizer combined. The soil organic carbon increased. So our work will focus on the following 4 hypotheses. The first one is, does the trend of increase in soil organic carbon observed with field experiments reflect the truth in the paddy ecosystems? Because for the long-term field experiments, the treatments are artificial or the ideal conditions, but compared to the farmers' conventional measurement, is this also increased in SOC. And the second is the carbon source or the carbon input from the ecosystems sufficient to support long-term carbon sequestrations. So if the carbon source cannot supply or offer the carbon sequence, so this will not last for long-term carbon sequestration. And the third, just at the beginning I mentioned, because of the wet, dry, or anaerobic and anaerobic cycling, so this condition, the paddy soil develop with biochemical functions which are conducive to organic carbon sequestration. And the fourth hypothesis, because we know in soil there are much autotrophic microbes, and the autotrophic can autotrophic fixation CO2, and is there any other carbon assimilation pathway in paddy soils? This means the autotrophic microbe can fix or synthesize CO2 into organic soil organic matter. So for the hypothesis 1, we monitor or take a sample from 4 different land scale in subtropical regions, and also we take 60 field sites managed by farmers to normal rice production. So we monitor the SOC changes or the SOC dynamics at the landscape scale and the farmers' conventional management. So we selected the paddy and other land use soil samples from low mountain, lowland, low hill, and karst mountains, and then measures the SOC content. So from this result, we can see under the different land use compared with the arable orchard woodland use, the spatial distribution of soil organic matter, soil organic carbon in paddy is higher than other 3 land use. This is the landscape scale. And also we see the SOC probability from the 4 different land use. This also sees the soil organic carbon content of the flower layer in the paddy are much higher than other 3 land use at the this 4 landscape scale. And also we compared during the last 30 years. We compared from 1980 to 2003 the changes in soil organic carbon content in a low hilly area, also in the paddy, are much higher than the upper land soil. So for the second hypothesis, so is the carbon input or the carbon source can support the long-term carbon sequestration from the rice ecosystems. So we, to test this hypothesis, we focus on the rice deposition carbon and what the contribution of the rice deposition carbon is derived from photosynthesis carbon of rice. We use the C13 C13 dioxide continuous or plus labeling to qualify how much carbon input into the soil organic carbon from rice reproduction carbon. So here we from the FAO database and the China statistics report data, we see that both in the total biomass or the yield of rice are increased due to the variety, higher yield variety use, and the double rice measurement, and the chemical fertilizer. The productivity of rice are increased. So this means during with the rice production, the rice straw or rice root input the soil are also increased as the meantime. So here we use the carbon 14 CO2 continuous labeling to qualify the how much rice photosynthetic carbon to the different carbon pool. Here we selected the 4 typical paddy soils, and we qualified that about 4 to 6% of rice deposit carbon into the soil organic carbon, and they are also there to the dissolved organic carbon, microbial biomass carbon, and other carbon pools. And we also found the missing effect from the rice deposition carbon input. So it means when rice grows the different soil organic carbon content. So from the higher, when the rice grows in higher soil organic carbon, the rice deposit carbon means the slow microbial biomass and the slow turnover and the higher contribution to the soil organic carbon. And on the contrary, when the rice grows in the lower soil organic carbon, they are about half of the rice deposit carbon lost by the respiration and result in the lower contribution to the soil organic carbon. And also we also compared the rice planted soil and no rice planted soil. The rice deposit carbon input to the soil resulting in the negative priming effect compared with the no rice planted soil, and this means the legacy effect of the rice deposit carbon will decrease the native soil organic carbon decomposition and will result in higher accumulation of soil organic carbon compared with the no rice planted soil, and this will also enhance the soil organic carbon sequestration. And this is the third, to test the third hypothesis, we are focused on the soil biochemical functions. We take following 3 sites to test the soil biochemical functions. The first one, we have a look at the size of the microbial biomass carbon in the soil and compared with the other land use. We take the huge samples and measure the microbial biomass carbon. We found the means of the microbial biomass carbon in paddy has 2 or 3 times higher than other 3 land use. This means the size of the microbial and the communities are larger than other land use. So also we use the carbon-14 to test the turnover rate or turnover time. We found for the turnover rate in the paddy are much higher than the upland soil, and the result in the faster turnover of time in paddy than upland. So this means we also calculate the turnover time converted to the field conditions. For the paddy, they're about 2/3 year, and for the upland, they are more than the turnover time. They are more than 2 years. This means the faster turnover in paddy than in the upland. And meanwhile, we compared the liable carbon, and we use the carbon-14 glucose and carbon-14 rice straw to monitor the mineralization. We found that in the paddy soil, they are, compared to the upland soil, both the glucose and the rice straw are slower than in the upland. And meanwhile, for the native soil organic carbon, they are also found in the paddy. The both the strong and the rice addition will not increase the native soil organic carbon mitigation in paddy. However, for the arable or for the upland, the new carbon input will significantly increase the native organic carbon mineralization for the arable soil. This means for the paddy, there exists a stronger positive pollen effect for the upland soil, and however, there is no or very weak pollen effect for the paddy soil when the new carbon input. And the fourth, the last hypothesis, we will qualify the autotrophic microbe in soil and how much autotrophic microbe can fix the CO2, and what is the contribution of the autotrophic microbe to the CO2 fixation. We selected 8 typical soils and uses the carbon-14 continuous labeling to qualify the autotrophic CO2 fixation. And meanwhile, we also use the molecular tool to qualify the functional gene numbers and their structures, and found what is a functional microorganism can participate in autotrophic fixation. So here we show the incubation experiment in a closed chamber to qualify the assimilation of CO2 by autotrophic microbes. So here we see during the 80 days continuous labeling, we found a significant C14 labeled SOC and C14 microbial biomass was determined, and we also estimate the assimilation CO2 rate is about 100 to 500 kilogram per per hectare per year by the artificial traffic CO2 fixation. And this amount is equal to the 10% of rice photosynthesis carbon input. And also we use the qPCR to qualify the functional gene abundance. There are huge functional gene copy numbers are bacteria, blue-green, and non-green CBBL copy numbers were found in our tested soil. And also the CBBL from bacteria are much higher. It's about 100 times than blue-green and non-green CBBL gene numbers. And this means the autotrophic CO2 fixation, main driver by bacterial autotrophic microbe to participate in this process. And also, we qualified the 4 different carbon sources. They're from rice shoot, rice root, rice deposition carbon, and our CO2 autotrophic microbial carbon. And we found that the greater contributor of the microbial CO2 autotrophic microbial are the greater contribution, the relative greater contribution to the other 3 sources. And the last, we also know there's the greater contribution to the methane emission from the paddy. So how to cut the methane emission from the paddy? So we recommend one measure to upland shift incorporation of rice Xiaojuan. So we all know if we return the rice Xiaojuan directly to the paddy, there will result as a huge methane emission from the paddy. And if we burn the Xiaojuan, that will result in the nitrogen loss, the smoke pollution. So how to management the rice Xiaojuan to both the win-win effect. So we took the field experiment for upland shift incorporation of rice Xiaojuan. We moved to the rice Xiaojuan to the labor vegetable field, tea, orchard garden soil, and this will improve the soil fertility, nitrogen loss control, and also reduce the methane emission. So we took up the long-term rice Xiaojuan incorporation in the paddy or in the paddy upland location, or also the low slope hill upland. So we, from the 10 years experiment, when we move to the rice Xiaojuan to the labor upland soil, the soil organic carbon increased compared to the chemical fertilizer, and also the total nitrogen and the total phosphorus also increased. And for the sweet potato and rapeseed yield also increased by 5% and 10% respectively from the 8 years rice Xiaojuan incorporation. And last, for the conclusion, so from our test to the 4 hypothesis, we can say that because of the increasement of the rice production and the result of the rice straw rice root increase into the paddy can support to the long-term sequestration. And also compared with the upland, there are higher microbial biomass carbon, faster turnover, low flash, and native soil organic matter mineralization, and the result is the negative or the weaker polyming effects in paddy soils, result in the higher carbon sequestration to the paddy. And also there is the significant amount of the autotrophic microbe to fixed CO2 in paddy can also support to the carbon sequestration. And the last, we found we take up a possible measurement to cutting machine emission. That is the move to the rice straw to the upland to cutting machine emission and improve the nutrition and control the nutrition loss. And at the last, I will thank our collaborators and our grantors to support our work. That's all. Thank you.
Thank you, Dr. Ger. We have a question that came through on the chat. The question is, in your paddy rice field sites, is the water management alternate wetting and drying or a single water drawdown each season?
Yeah. Thank you for your questions. Yes, as we mentioned in the in my talk, the water content or the dry wet organism, this will result in the redox changing, because we know for when the flooded, the paddy flooded at the very wet condition, this means the redox is lower and the oxygen is limited. And for the oxygen limited condition to decomposition of the soil organic matter, they need less energy, and the microbial biomass carbon turnover is slower, and maybe will result of the net net carbon sequestration compared to the upland.
Thank you. I think your answer has covered, there was a second question about the effect of water regime on soil carbon flux, and I think you just answered that question right there. So thank you very much for your presentation. Thank you. Our next presenter is Dr. James Tida. He's a university distinguished professor at Michigan State University, a member of the U.S. National Academy of Sciences, and a foreign member of the Chinese Academy of Sciences. He studies microbial ecology, physiology and diversity, biodegradation of environmental pollutants, and use of molecular methods to understand microbial community structure and function. And Dr. Tida is going to offer comments as a summary for the presentations tonight. Jim.
Okay, thank you very much. And I really enjoyed the presentations tonight. So in summary, I would say that we had two types of presentations. Two that were very global, showing how carbon management really is important at the global scale, and two that are more field experimental and mechanistic based studies. So both are important in understanding how to manage carbon in the soil for better outcomes for our planet. Just a few things from each presentation. Rattan Law talked about several things relating to this larger scale and used terms like sustainable management, recarbonization of the terrestrial biosphere, carbon farming, societal values of soil organic carbon. And he proposed a timetable with 6 possible types of changes that are possible if people would put their efforts in those directions that would help us achieve the goal that we strive for by reducing the carbon contents in the atmosphere. And concluded with his mantra that healthy soils makes everything healthy. Everybody, everything, the planet. Then Professor Feng had a very detailed mechanistic study in the grasslands of the alpine regions with some very sophisticated marker studies and understanding about carbon turnover in these experimental systems. Talked a lot about carbon use efficiency and microbial necromass and how that's an important part of the carbon cycle. Then Professor Grunwald also talked on the large scale and showed us how models are going to be an important part of understanding and managing carbon. So data and sensor-driven approaches. She talked a lot about the use of AI and its various subcomponents like machine learning, deep learning, and different AI models. So I was quite impressed by the improvements in understanding carbon using AI. So this is a data-driven approach as opposed to a mechanistic approach. And she showed us in some cases where the data-driven approach using AI is seemingly more accurate, sometimes values of greater than 0.9 in terms of their accuracy. So that's good, but we still need the mechanism understanding as well. Then the last presentation by Professor Ger, again, was an experimental field-based study. He tested 4 hypotheses related to the rice paddy system. Showed us an example of negative priming, that the turnover time of carbon in the paddy was less, like 0.7 per year versus the upland 2.2 per year. Also showed us how important autotrophic autotrophic carbon fixation, some of that's also nitrogen fixation, there is in the paddy soil as well. And he also pointed out the problem of methane production in paddy soils, especially if you add the rice straw, and then proposed that moving the rice straw to the upland situation was a much better solution to the problem of methane production and also benefited the upland productivity. So those are my summary points. I thought it was a very good set of presentations, and especially because it covered a broad range of approaches, systems, and experimental and more global. So thanks very much to the speakers tonight, or today.
Both, actually, which is fantastic. Yes, thank you, everyone. On behalf of Feng Wang, who said, yes, make sure we thank everybody. Yes, thank you, all of you speakers and panelists and moderators and the organizers and just the immense amount of teamwork that put this event together. Thank you for attendees for hanging out. And I understand we have about 3,000 people viewing this in total right now. So fantastic. And yeah, stay tuned for the next one. The idea is that this is the first event to kick off this joint effort. So watch your respective society, whether that be in the United States, well, in America/North America and other Americas and elsewhere, and China and everywhere else that we have our members and our colleagues in this soil science field. So Thank you, thank you, thank you, and enjoy the rest of your week or evening or day, whatever that might bring when you are viewing this. So thank you again, and goodbye.
Thank you. Thank you.
Thank you.
Bye-bye.
Thank you.
Bye.
Bye-bye.
Thank you. Bye-bye.
Most welcome. Goodbye.